Once again, thank you very much to PSM for inviting me to be a part of this activity. I'm really very grateful to be speaking to the 300 professors and students who have been invited to be a part of this event. Once again, I am June Barnes, and I am working for 3M Philippines as its professional service. Now, before I go to my talk, I just want to reflect on some of the things that were pointed out by Professor Carlos in her talk earlier. So earlier, Professor Carlos talked about emerging foodborne diseases. And from her talk, we are actually faced with two realities when it comes to foodborne pathogens. First is the fact that these pathogenic microorganisms can pose safety risks, and the fact that these microorganisms can also change. Change is a fact of life. But of course, with the microorganisms changing, for us microbiologists, that can be quite a challenge for us because methods that were previously developed to detect a specific pathogenic microorganism may suddenly miss that pathogen as a result of that change. And as you know very well in the food industry, missing this foodborne pathogenic microorganism is something that is very risky and would have serious consequences. And I really believe that it is because of these realizations by the food industry, that's why a lot of laboratories of these food manufacturing companies are now looking for alternative tests that would meet their needs. And that's why for this morning, I will be talking to you about 3M's, modern microbiology methods for the detection of important foodborne pathogens. And for our session this morning, I'm going to share with you why the food industry is testing for pathogenic microorganisms, some of the pathogenic organisms that they are testing, as well as the challenges that they are facing in their laboratories. I'm also going to cover a few of these methods which are being used in the food industry, and I will be ending our discussion with some criteria that you can use in your selection of a pathogen test. But before I begin my talk, I just want to emphasize a few things. And one of them is the fact that I have been working very closely with the food industry for the past 15 years. And that is why majority of what I will be sharing with you are based on my observations and interactions with key players in the food industry. Secondly, as you might have noticed, I did not go directly to the methods, which is my actual topic, because I want to have some time to provide with you some insights as to why the food industry is looking for alternative methods. Bakit hindi sila nakokontento na yung conventional agar na lang yung gamitin nila? And lastly, some of the methods, if you look at it very carefully, may not really be very modern in principle, but it is something that is still considered modern by our local food industry, or it may be an old technology, but it's modernized by a supplier. So I just want to make all of these things clear before I begin my talk. So let's go to question number one. Why test for pathogens? For food manufacturing companies, testing for pathogens is really very important in order for them to comply with regulatory and customer requirements. So for example, if I am a meat manufacturer, before I can sell my product, it's really very important for me to satisfy the local regulatory requirements that would be the Food and Drug Administration before I can sell the product. And normally part of the criteria would be screening for pathogenic microorganisms. Now, it's also possible that I may be selling my raw materials or my ingredients to a toll manufacturer. So before I can pass as a supplier, part of the requirements of these food manufacturers would be testing for pathogenic microorganisms. So regulations and customer requirements are some of the drivers why food manufacturers need to screen for pathogens. But of course, another reason why food testing needs to be done, screening for pathogens need to be done, is because these food manufacturing companies need to ensure safety of their customers. They need to make sure that their products are free from pathogenic microorganisms before they are sold to the market. And of course, it's really very important for them to do this in order to protect their brand. You don't want to be involved in any outbreak or any recall only because of a contaminated product. Pero ako, being part of the food industry for the past 15 years, I'm really very proud on how yung ating local food industry is advancing when it comes to food safety practices. A lot of these food manufacturers, whether it's multinational or a local company, are actually including now pathogen testing as an important element of their food safety plans. And fact is, they are investing a lot in order to develop the capability to perform it. Now, just to share with you another observation, unlike in the past wherein these food manufacturers would really just focus on end product testing, that practice is actually changing. We have observed that many of them would also do controlling of the microbiological quality of their raw materials and the incoming materials, as well as the environment. That's why we see a lot of pathogen testing happening in these areas now. So hindi na lang tayo limited sa end products, the raw materials and environmental samples are also screened for pathogenic organisms. So what pathogens are these food manufacturers screening? Well, the choice of pathogen or the pathogen of concern really depends on the food matrix that is being manufactured. And I actually presented here a table of the different market segments and the pathogens of concern. And if you will notice in the case, for example, of dairy, Salmonella enteritidis, Salmonella Staphylococcus aureus, Listeria, or Listeria mono, Salmonella, and Chronobacter would be the pathogens of interest. A lot more as compared to the seafood industry, for example, which is a lot more concerned with Vibrio, Listeria, and Salmonella. Now, if you're going to take a look at all the market segments, you might have noticed that the dominant pathogen of concern is really Salmonella. And that is why if you take a look at the pathogen testing market, it's not surprising that almost half of the 183 million pathogen tests conducted is really focused on Salmonella, followed by Listeria. And only a very small chunk of the pie is allotted for other pathogens like Campylobacter and E. coli 0157. So if you're into research and you want to develop commercial kits, it's really Salmonella testing and Listeria testing, which is the way to go because that's what the market needs. I also want to share that when it comes to allotment of samples, the share between food samples and environmental samples would also vary depending on the pathogen of concern. So if you're going to take a look at the table for Salmonella, you might have noticed that more samples, food samples are tested as compared to environmental samples. But in the case of Listeria, more environmental samples are screened as compared to food samples because Listeria is the environment is a more significant source of this pathogenic microorganism. Now, I also want to highlight that our local FDA provides very clear definition as to the food products that should be tested for these pathogens. And if you're going to take a look at the revised guidelines for the assessment of microbiological quality of processed foods, you might see there that for Salmonella, there is a total of 45 food matrices that need to be screened for these pathogen, 10 food matrices for Listeria monocytogenes. For coagulase positive Staphylococcus aureus, there are 28 food matrices, and there's also a few matrices that would require screening for Campylobacter, Bacillus subtilis, Bacillus cereus, Clostridium perfringens, Chronobacter, and Vibrio parahemolyticus. And I also want to highlight that sometimes a customer would require pathogens on top of the regulatory requirements. Like, for example, there is a local toll manufacturer here wherein they would ask their suppliers to also screen for Clostridium difficile. Now the question when it comes to testing is it easy to test for pathogens? Based on your experience, ano pong masasabi ninyo? Well, if you're going to ask microbiologists that I have encountered in the food industry, most likely they will tell you that it is not very easy. And the reason for this is because of the low prevalence of pathogens in food samples. And another challenge is just in case they are present, they are normally present in very low numbers, and their distribution is often non-homogeneous. So imagine finding Salmonella in 10 tons of processed meat. It can be quite a challenge for the food manufacturer. We also have to recognize the fact that it's possible that the test or the target organism may be inhibited because of inhibitory substances that can be found in the food sample. And this is very true in the case of difficult to test samples like spices, garlic for one, very challenging to test, as well as environmental swabs or sponges because of the residual disinfectant that may mix together with the target organism. Now, another challenge that's faced is that the laboratory should be able to ensure that they are able to detect even just one colony forming unit of the target organism in 25 grams. So they need to make sure that the method that they have chosen is sensitive enough to screen for this a very low number. Now, on top of this, the reason why a lot of food manufacturers are looking for alternative methods is because of challenges when it comes to operational side. And one of the greatest concerns of these food manufacturers would be time to results. If you're going to follow the traditional method, BAM, for example, normally you would take about 150 hours before you can conduct biochemical confirmation. And for food manufacturers, 5 days, that's just way too long. Remember that for food manufacturers, time is money, and they want to release the products as quickly as possible. This can also be a challenge for food manufacturers wherein their product has short life. Imagine a product with 7 days only of shelf life, and I need to wait for more than 5 days to get the results. So by the time I get the results, the product is already expired. Of course, there's also a need to develop expertise when it comes to interpretation of colony morphology on agar media. If you take a look at the interpretation guides and books, plates would often look very perfect, something like this, as if identification of the target organism is very easy. But when you work in the food industry, it doesn't always happen like this. You will be required to test for raw materials with very high microbial background. And the consequence is the presence of these other organisms may actually interfere with your target organism's growth and may also pose a challenge on interpretation. Ang hirap ma-identify ng target organism. That is on top of the need to develop the expertise for the interpretation of colonies of atypical organisms. Let's have Salmonella as an example. This is Salmonella enterica subspecies enterica. This organism would have these characteristics. That's why when you grow them on media recommended by BAM, this is how the organisms would look like. But we have to recognize that there are different strains which would show different colony morphologies. This is still Salmonella. This is still Salmonella. So imagine having an analyst which does not have a very strong background or experience on Salmonella testing, then it's possible for them to report it as negative because they are not familiar with the atypical strains, when in fact, the organism is present in the sample. And this is also true in some media, which has the tendency to provide a lot of false positives. And I'll go to Salmonella as an example. As I've mentioned earlier, Salmonella would produce hydrogen sulfide, but they cannot ferment lactose. So when you grow them on XLD, this is how the organism would look like. But we have to recognize that other bacteria like Citrobacter and Proteus would also have the same characteristics, and thus you will form colonies similar with that of Salmonella. And that becomes a problem because you would do further tests to your sample, even though what you have really is Citrobacter and Proteus and not Salmonella. And that adds up a lot of time before you are able to prove that Salmonella is present or absent in your sample. I'll probably skip this because I am running out of time. I want to go now to false negatives, which I believe is an even bigger challenge in the food manufacturing industry. And the reason why is because you would think that the pathogen is absent when in fact it is present in your sample. And this is a big challenge because some microorganisms, some pathogens have been reported to be viable but non-culturable, which means that they are present in your sample, but they lost the ability to form visible colonies on agar. And the consequence of that is you will not see any colonies, therefore you will report it as a negative, when in fact you are positive for the organism. And this is very dangerous in the food industry because there has been a report that says that exposure to chlorine at levels which are normally used in the food industry, they can actually make pathogenic organisms like Salmonella and Listeria monocytogenes viable, but not culturable. And that would increase the risk of missing these organisms. So if you combine all of these challenges, I believe these are the reasons why a lot of food companies now are moving from traditional agar methods to more modern methods that would allow them to get the results faster without sacrificing the specificity and sensitivity of the assay. And down that leads me to question number 4. How do I test for pathogens now? Okay. Well, our generation is very lucky because we have a lot of choices now when it comes to pathogen testing, but it actually did not happen quite fast. Pathogen testing has definitely gone a long way when it comes to detection of pathogens, but again, it didn't happen quick. It actually took a lot of years. If you will notice, there's a huge blank space before an alternative method was developed in 1971, and that is through an alternative method called immunoassay. The good thing is after the next 50 years, there's a lot of improvements when it comes to technology change. And some of the technologies introduced would include the PCR technology. PCR stands for polymerase chain reaction, as you know very well, and this was developed by Carrie Mullis in a laboratory in California. And because of this, she actually won the Nobel Prize in Chemistry in 1993. Now, after 14 years, there's a new molecular method that was introduced in Japan, and that technology is called isothermal DNA amplification. And I'm presenting these to all of you because these are the technologies that you will commonly find now in food companies. And I will be sharing with you these methods as we go along with our discussion. Okay. Well, of course, our discussion today, although we want to focus on modern technologies, it won't be complete without having a review of traditional methods because we want to have a good understanding of what makes each one of them different from one another. I'm sure that all of you are very familiar with this. You start with your sample and then you subject it to enrichment. Normally, you have your primary enrichment and a secondary enrichment. Afterwards, you subject the enriched samples to plating so that you will be able to detect the target organism. Once you have detected typical colonies of your target organism, that is when you subject the colonies to confirmation. Now, in the case of alternative method, it would also have 5 main parts, as you will see here. You start with your sample, then you do your enrichment. But the difference now is that you would have some type of sample preparation that would allow you to have enhancement to facilitate better detection. So after your sample prep, that is when you do your detection, which can be molecular based or immunological. And once you get the result that is positive, those results are considered presumptive, and thus you need to do your confirmation. Now, based on the 2 slides, you might have noticed that regardless if you are using a traditional method or an alternative method, one essential step would be enrichment. And the reason for this is because pathogens would normally be present in low number in your food sample, or they may be stressed because of all the processing that the food needs to undergo. And because of this, it's really important for us to do enrichment so that we can recover injured or stressed cells, and at the same time, increase the number of the target organism to a detectable level. And during enrichment, it's really important for us to use the appropriate medium as well as conditions that would create an environment where your target organism, that would be the red one, would be enhanced. So you do that by inhibiting the non-targets and allowing further growth of your target organism. So by doing this, you will be able to enhance the detection of your target organism. Now, if you have a review of the different protocols available now, you might be surprised that they would have differences in terms of the number of enrichment steps. Now, generally, in the case of traditional agar and immunoassays, you would have 2 to 3 enrichments. And you normally start with your primary enrichment using a non-selective medium. And this is followed by a selective enrichment so that you're able to select the target organism and remove the competing microorganisms. Now, in the case of immunoassays, I have encountered some methods wherein you will have a post-enrichment step. One very good example would be in the case of Salmonella. For immunoassays, you would have post-enrichment using M broth. And the reason why they added this is because it enhances flagellation, which is important for increasing the antigenicity of your test, of your target organism. Now, this is where the advantage of molecular methods come in, because normally for molecular methods, you can just have a single enrichment. And the reason for this is because these molecular methods are actually able to seek out the organism, the target organism's DNA with the use of primers and probes. And all you have to do here is just make sure that the number of organisms would be at a detectable level. Now, with regards to traditional testing method, you know very well that our principle here is really just forming the colonies and screening the colonies, because those colonies will tell you a lot of information in terms of its biochemical characteristics. In many of this media, you make use of different chemicals in order for you to have an idea on the organism's metabolic characteristics. So you would add antibiotics to screen for ability to tolerate it, or sugars to check utilization of the organism by the organism. Now, here in the Philippines, we are a BAM testing country. So majority of our customers would follow BAM, which is the preferred method of many US FDA laboratories. But for food manufacturing companies here in the Philippines, which are European, most of them would follow the ISO standards. And I'm happy that in recent years, there has been a lot of advancements also when it comes to traditional methods. They have now incorporated chromogenic substrates in a traditional agar in order for you to have a better differentiation of your target microorganism from background flora. And right now, it's a lot more convenient because ready-to-use plates are also available. So you can buy pre-poured plates, or you can make use of rehydratable film plates. So whenever you do need to do the test, just take it out from your storage and do your testing after enrichment. Now, just to give you an idea, here is one example of a chromogenic agar. This is Ramback agar used for Salmonella detection. So this medium would make use of a chromogen that would screen for, or that would indicate a beta-galactosidase activity. Remember that coliforms would have that ability, and the consequence of that would be formation of blue-violet colonies. So these are your coliforms. Now, in the medium, they have also incorporated propylene glycol, and that is where Salmonella would, that's what Salmonella would utilize, causing a drop in the pH. And because of that, Salmonella would have the characteristic pink to red colonies. Quite easy to use as compared to your traditional agar. Now, this is an example of your ready-to-use plate. They are prepared for you. And normally they would have a shorter or more streamlined processing. And in this case, this is 3M Petrifilm Salmonella Express system. An advantage of this one is that this is where you do your primary enrichment. And this is also where you do your confirmation with the use of a confirmation disk. But again, many laboratories now would move from traditional to other methods. And these other methods would include immunoassays and molecular methods. I'm sure you are familiar with immunoassays. They are based on antigen-antibody interactions. And the two most commonly used formats would be ELISA and lateral flow devices. But more now are using molecular methods as a major development. And in molecular methods, you target specific genes of your pathogen of concern. And the specificity is dictated by the probes and the primers which are being used. Now, just to give you an idea, let's go through each one of them, starting with your immunoassays. Since we're talking about developments, I just want to stress that aside from these two formats now, there are two other formats available called the alpha as well as the immunodiffusion. Now, for the first three, they would make use of conjugates, which are basically antibodies specific to the target organism. But what makes them different is that they are labeled with a marker. And these are some of the markers commonly used. Now, just to illustrate how these work, let me just present this to you. So this is how a lateral flow device would look like. If you take a look at the content of these tests, you would see there are 4 sections. You have your sample pad where you would put your sample, and then it will pass through your conjugate pad. That's where you have the antibody with the marker. You then have your nitrocellulose membrane, is where you will find 2 lines, your test line and your control line. And at the end, that's where you have your absorbent pad in order to allow migration of your sample through capillary action. Now, since I know there are students here, I made some cartoons for you to imagine what happens. So this is the cross-section of your test. You have your sample pad, you have your test line with the antibody specific to your target pathogen, and you have the control line, which has the antibodies for the other antibody. Now, in your conjugate pad, that's where you have your antibody for your target pathogen, but it's linked with your marker so that you have an indication if there's an interaction between your sample, your target pathogen, and the antibodies. So let's imagine first having a negative sample. So once you put your sample in the pad, you now have your conjugate here that will start the migration. Now, since there is no sample to link your conjugate and your antibody, as the liquid flows, it will just continue to flow. Now, remember that this antibody can recognize the antibody that you're using. So consequently, they will be linked together. So once you check the result, this is what you will see. You only have 1 line, meaning you are negative for the test. Now, in the case of positive samples, the moment you put your sample, it will now bind to your conjugate. Now, remember that this antibody can also recognize this antigen. So the moment it migrates, there will be linkage between the two, so you're able to form the sandwich. The rest of the conjugates will now flow until it reaches your control, and they will establish a relationship between the two. So when you check the results, this is what you will see, 2 lines. Now, ELISA basically follows the same principle, except that you make use here of microwells, which are coated with antibodies. So here, after enrichment, you put your sample, and if you are positive, the antibodies will bind your target antigen. So you'll do extensive washing. This step is called capture. You then add your conjugate, and in the case of positive, you'll be able to form the sandwich. This is what we call conjugate binding. Very important for you to do extensive washing prior to adding the substrate. Since you have the enzyme here, it will react with your substrate, causing color change. And that color development will tell you that you are positive for the target organism. Now, a modification of that is what we now call the alpha. The nice thing about this is that you make use here of cassettes, and these cassettes would already contain all of your reagents. In the system, you also make use of a pipette with antibodies impregnated on it. We call this pipette as a tip, as your solid phase receptacle. So what happens is, as you press your pipette to the sample, if you have your pathogen, it will bind to your antibody. and then you will have a lot of washing so that any unbound microorganisms will be removed. It will then pass through the conjugate, where you will form this interaction, series of washing again to remove unbound conjugates, and lastly, you have the substrate, allowing you to form this sandwich. Now take note that the interaction of the enzyme with the substrate will now produce fluorescence, and that fluorescence will now be read by the machine. This is an automatic process, so it's very convenient for food manufacturers. Now, if you want a simpler process, we have immunodiffusion. You make use here of semi-solid media. One chamber is where you put your sample, and the other chamber is where you put the antibody. So during incubation, what happens is there will be migration of the salmonella and the antibody, and their interaction will be marked by a smiley face. And that smiley face would tell you that you are positive for the target organism. This process, however, becomes a challenge if you have non-motile target pathogen. Now, the last one, and I'll make it quick, would be molecular methods. And you target genes of your target organism. But unlike in research laboratories, wherein they often use open systems, in the case of food manufacturing companies, they prefer closed systems. And what I mean by that is they make use of the media, they would make use of the kits, and they would make use of the instrument and software provided by the developer. So you are limited to the availability of kits produced by the supplier. But for food manufacturers, this is something that they really appreciate because normally the process is a lot more streamlined. And not only that, often you don't have to do any reagent preparation anymore. And that increases their productivity in the laboratory. Now, the two most commonly used formats would be PCR, specifically real-time PCR, and a newer molecular method called LAMP, or loop-mediated isothermal DNA amplification. Let's go first to PCR, which is something that you are very familiar with. Of course, you need your DNA template, yung i-amplify mo. You would need the primers, which would be specific to the target organism. And of course, you need your polymerase. In the case of PCR, you make use of the Taq polymerase. And you know very well the process. You know the process very well. You let the DNA undergo denaturation, so there will be separation, annealing of the primers, and then you increase the temperature to 72 degrees Celsius so that you will have amplification. So if you started with one DNA, you will end up with two after one cycle. And the process will continue until you have multiple copies of the target DNA. And this is made possible, of course, with the use of thermocyclers. Now, in many closed systems used in the food industry, they would make use of real-time PCR. And the nice thing about this is that you're able to monitor amplification as it happens. And you normally make use here of fluorescent indicators. Specifically, they love using probes. One thing about probes is that when they are bound to your single strand, they don't fluoresce because of the quencher. But the moment the probe becomes displaced as you amplify the DNA, that is when fluorescence is expressed. So the more DNA you produce as a result of amplification, what happens is you generate a higher fluorescence signal. And that's actually very true in this system. So if you will notice, the process of testing is very simple, and it comes with the software. So interpretation of the results become very easy because interpretation is just done by the software that is provided by the supplier. So let me focus now on the LAMP technology. It's molecular, but it's very different from PCR. In what ways are they different? Again, in PCR, if you might have noticed, they only use two primers for the two strands. But in the case of LAMP, they normally use 4 to 6 primers that would recognize more distinct regions in the target DNA of the pathogen. And In the PCR, you need a thermocycler because you need 3 different temperatures. In LAMP, ISO 1, you only use 1 temperature, and that's 60 degrees Celsius. For PCR, the polymerase that you're using is TAC, but for LAMP, they're using BST polymerase. Now, in terms of detection of the amplification process, most PCR technologies would rely on fluorescence, but in the LAMP technology, it can be done using fluorescence or by the use of bioluminescence. So just to differentiate, these are the primers used for the LAMP technology. You have more. These are your primers used in PCR. Now, again, in terms of detection of amplification, it can be done using fluorescence. So in one product, they make use of a molecular beacon. This is complementary to the target DNA, but they are bound together. So fluorescence is still not displayed. Now, the moment you amplify the DNA can bind to your molecular beacon, and it will cause separation of the 2 ends. And this will now signal fluorescence, and that fluorescence will be read by the machine. Now, in the case of another product, they combined ATP bioluminescence with isothermal DNA amplification. And the principle behind here is really fireflies. Fireflies light up because of the luciferase enzyme. And for it to light up, you need energy. And that energy is supplied by ATP. So the moment they come together, that's when light is produced. So where does isothermal DNA amplification comes in? Well, it turns out when you amplify the DNA, you produce a lot of pyrophosphate. So the developer of the technology said, why don't we combine it with adenosine phosphosulfate in the presence of sulfurylase enzyme, which will serve as a glue. So what happens is this two will form your ATP. Because you have the ATP, once you have the luciferase enzyme supplied to the reaction, you produce the light. So light production means you are positive. No light means you are negative for DNA amplification. And the system also comes with a software. A peak like this means you are positive, and a flat line like this means you are negative. And the nice thing about the use of bioluminescence, which is something that is relatively new, is that you can monitor amplification real time. You don't need to spend on expensive fluorescent dyes and fluorescent detectors. And it has also been shown that ATP bioluminescence can actually be more robust and reliable when it comes to food testing, because fluorescence can be exhibited by certain food products. Again, because we're using fluorescence, it's now possible for us to use color coding, which is not possible in fluorescence. And also because of this, it's now possible for us to just develop one protocol for all pathogens, which means that you can do testing of all these pathogenic microorganisms in the same run, since the protocol for detection is the same. I really want to highlight these things, but I'm running out of time. And I just want to highlight that I think there's enough evidence to prove that the LAMP technology can be used for foodborne pathogen detection. And these are several articles pointing out the specificity and sensitivity of LAMP compared to PCR assays. And again, they highlight that the reason why you're able to achieve it is because you're using more primers for specificity, more primers, more specific. It's also because you're using a different polymerase, which is a lot more robust compared to PCR, the Pac polymerase, which can be inhibited by certain compounds found in foods and environmental samples. And I think in terms of cost, because you don't need to have a thermocycler, in terms of investment, it's a lot lower for food manufacturing companies. I also want to highlight that there's another article that points out a sensitivity of the method. And they said that LAMP has higher specificity, higher sensitivity than PCR. And these articles even point out that it's 100 times more sensitive than PCR. In the article, They also compared LAMP technology with PCR. And if you will notice here, they also pointed the advantages and limitations. And there's just way more advantages of LAMP technology for foodborne pathogen testing as compared to PCR, which has a lot more limitations. And this is another article. This is basically their conclusion. They said it's an innovative technology for food safety testing, which has high sensitivity, specificity, and rapidity, and that it's a lot simpler because you really don't have to do a lot of sample preparation. Now, to end, I do know that there's already a lot of methods available in the market, but there are some criteria that you can use for selection. Of course, speed and ease of use would be a priority for the food industry, but there are other criteria which I believe are more important, like for example, equivalence. It needs to perform equivalent to our standard methods. You also need to be accurate. So doing verification prior to adopting the method is really very important. You definitely don't want any cross reactions because you don't want thinking the pathogen is there, but what you actually have is not your target pathogen. So you want it to be very specific. And of course, consider the limit of detection because the lower the number of organisms that can be detected, that can be better. So we always recommend to our customers to ask for validations of these products because these validations would prove the performance of these test methods. I know I'm showing here three. These are the commonly encountered validations, but among these three, it's always best to go for official methods of analysis as well as AFNOR or microval, simply because more laboratories tested the product and there are also published studies compared to PTM, wherein testing was only done by one laboratory. As a summary, I think I was able to highlight that these pathogens are very important hazards in the food industry, and that's why testing should be done. But with traditional methods, there's a lot of challenges that the food industry is facing. It's a good thing that nowadays there's a lot of methods available for you. Now, to end, I just want to share this because whenever I give talks like this, participants would always ask me, can all of these test methods detect the pathogen? Well, the answer is actually yes, they can all detect the pathogens, but we have to recognize that they have different sensitivities and specificities. And the reality is, if you are making use of a method that is not sensitive enough, and the other body, like for example, a regulatory body is making use of a better technology, there's always a big chance of you missing it and someone else finding it. And I really challenge food manufacturing companies that if you want to avoid this, if you want to avoid being part of food recalls, which is actively being done particularly in the US, it's really important for you to make use of a technology that can be very sensitive and accurate because it is worth the investment. I'll end my presentation with this. I'm sorry if I extended a bit, but just in case you want to ask further information, you can email me or message me in this number. Yun lamang po at maraming salamat sa inyong lahat.